Hi, welcome back to Daily Lectures by New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. We're on the subject of molecular biotechnology, and in particular on Chapter 8 of that deals with directed mutagenesis and protein engineering. Right, and we're on the topic of oligonucleotide-directed mutagenesis with M13 DNA. Oligonucleotide-directed mutagenesis, specific mutagenesis, is a straightforward method for producing defined point mutation in its clone gene, figure 8.1. For this procedure, the investigator must know 1. the precise nucleotide sequence, which has already been mentioned, and I'm just mentioning the first 3 points. And in the region of DNA that encodes the messenger RNA, mRNA, the amino acid changes that are being introduced, and in the early version of this method, the clone gene was inserted into a double-stranded form of DNA, form M13 plus strand of the recombinant vector was isolated and mixed with a synthetic oligonucleotide. The oligonucleotide had, except for one nucleotide, the sequence exactly complementary to a segment of the clone gene. The nucleotide difference, example that is mismatch, coincided precisely with the nucleotide of the mRNA codon. Now, the oligonucleotide hybridizes to the complementary region of the clone gene if it is added in an amount much in excess of that of the M13 DNA. If the mismatch is near the middle of the oligonucleotide, and if the mixing is done at a low temperature in the presence of a high concentration of salt, the 3' end of the hybridized oligonucleotide acts as a primer site for the initiation of DNA synthesis that uses the intact M13 strand as the template. The replication, which uses the deoxyribonucleoside triphosphate is catalyzed with the clonal flag fragment of E. coli. DNA polymerase 1T for DNA ligase is added to ensure that the last nucleotide of the synthesized strand is joined to the 5' end of the primer. Since in vitro DNA synthesis is often incomplete, partially double-stranded M13 molecules must be removed from the mixture by sucrose gradient centrifugation. And the double-strand M13 molecule, now containing the mismatched nucleotide, is introduced into E. coli cells by transformation. The infected cells produce M13 virus particles, which eventually lyse the cells and form plaques. Theoretically, because plasmid DNA is replicated conservatively, half of the phages that are formed carry the wild type sequence, and the other half contain the mutated sequence that has the specified nucleotide change. Phages produced in the initial transformation step are propagated in E. coli, and particles that contain only the mutated gene are identified by DNA hybridization under highly stringent conditions. The original oligonucleotide containing the mismatched nucleotide is the probe in these hybridized hybridization experiments, and will bind only to the mutated gene under these conditions. After the double-stranded form of M13 is isolated, the mutated gene is excised by digestion with restriction enzymes and then spliced onto an E. coli plasmid expression vector. Before we proceed any further, let's thank the authors Jack and Cheryl, Bernard, Jack and Cheryl, and the editors of the book, and for all those contributors, thank you and courtesy to all of them. Please do not, to all candidates listening to my lectures and taking screenshots, please do not commercialize any portion of my lectures or what you see on your screens. Thank you for your time listening to me. Thank you for your cooperation, and I wish you all the best in your studies. This is a diagram which represents the oligonucleotide-directed mutagenesis, single-stranded bacteriophage, M13 plus strand carrying a clone gene is annealed with a complementary synthetic oligonucleotide containing one mismatched base. That is one base that is complementary to its counterpart in the target DNA, with the oligonucleotides as the primer. DNA synthesis is catalyzed by the clonal fragment of E. coli. DNA polymerase 1. The clone gene and the M13 vector are the template. Synthesis continues until the entire strand is copied. The newly synthesized DNA strand is circularized by T4 DNA ligase. The ligation reaction mixtures is used to transform E. coli. Both the target DNA with its original sequence and the mutated sequence are present in the progeny M13 phase of DNTPs, deoxynucleoside triphosphates. Further study the altered protein is expressed in and purified from the E. coli cells. In actuality, when the oligonucleotide-directed mutagenesis is used, the expected 50% of the M13 viruses carrying the mutated form of the target gene are not recovered. Rather, for a variety of technical reasons, only around 1% of the blocks actually contain phage carrying the mutated gene. Consequently, the oligonucleotide-directed mutagenesis method has been modified in several ways to enrich for the mutant phage blocks that can be obtained. For example, one approach is to introduce the M13 viral vector carrying the gene that is to be mutagenized and into an E. coli strain that has two defective enzymes of DNA metabolism. One enzyme is a defective form of DUTPH, cells without a functional DUTPH have an elevated level of DUTP, which in turn causes a few DUTP residues to be incorporated into DNA during replication instead of the DTTP. The other enzyme is a defective uracil N-glycosylase UNG. In the absence of functional uracil and glycosylase, the DUTP residues that were sparsely incorporated into DNA cannot be removed. The single-stranded M13 DNA that is produced by this E. coli strain has approximately 1% of its thymidines replaced by urines. A mismatched oligonucleotide primer is hybridized to the substitution containing M13 DNA, and the second strand of M13 is prepared by in vitro synthesis and ligation. The double-stranded bacteriophage DNA is next introduced into E. coli strain that contains a functional UNG gene. The active uracil and glycosylase in the host cell then removes uridine residues from the transforming M13 DNA. As a result, the original M13 template strand is degraded, and only the mutated strand, which contains no DUTP is replicated. In this way, the yield of M13 bacteriophage carrying a gene with a site-specific mutation is significantly increased. Oligonucleotide-directed mutagenesis with plasma DNA. The major drawback to the bacteriophage M13 oligonucleotide-directed mutagenesis is the large number of time-consuming steps that need to be performed before the mutated form of the target gene is eventually isolated. As an alternative to the M13 system, a number of protocols that allow oligonucleotide-directed mutagenesis to be performed with plasmid rather than M13 DNA have been developed. With this approach, the need to subclone a target gene from a plasmid into M13 and then, after mutagenesis, clone it back into a plasmid is avoided. One of the plasmid-based mutagenesis protocols, the target gene is inserted into a multiple cloning site on a plasmid vector that contains a functional tetracycline resistance gene and a non-functional ampicillin resistance amp gene as a result of a single nucleotide substitution in the middle of the amp gene. The vector carrying the target DNA is transformed in the E. coli host cells to increase the amount of DNA through plasmid replication. Following growth of the transformed cells, the double-stranded plasma DNA is extracted and then denatured by treatment with alkaline solution to form single-stranded circular DNA molecules. 3 different oligonucleotides that anneal to one of the single-stranded circular DNA molecules are added to the sample of denatured plasmid DNA. One oligonucleotide is designed specifically to alter the target DNA. Another is designed to correct the substituted nucleotide in the non-functional ampicillin resistance gene. And the third is designed to change the single nucleotide in the tetracycline resistance gene so that the gene will become non-functional. The 4-dioxyribonucleoside triphosphates and T4-DNA polymerase, which has the same activity as the clinofragment of E. coli DNA polymerase 1, are added to the reaction mixture, and the 3' ends of the annealed oligonucleotides act as primers for DNA synthesis, with the intact circular DNA molecule as the template. The next in the synthesized strand are sealed by T4-DNA ligase. After synthesis and ligation are complete, the reaction mixture is used to transform E. coli cells. Transformants are selected for ampicillin resistance and tetracycline sensitivity with this procedure. About 90% of selected transformants have the specific mutation, the target gene, and the remaining transformants, the target gene is unchanged because the oligonucleotide did not anneal to the target gene, or it was bypassed during DNA synthesis. The cells with the specified mutation, the target gene, are identified by DNA hybridization. All the plasmids host bacterial strains, enzymes, oligonucleotides, other than the one needed to alter the target gene, and buffers for this a soulless kit, facilitating its widespread use. On to the milestone. Note: oligonucleotide-directed mutagenesis using M13-derived vectors, an efficient and general procedure for the production of point mutations in any fragment of DNA. The credits go to M.J. Zola and M.J. Smith. The technique for oligonucleotide-directed mutagenesis, site-specific mutagenesis, was developed mainly in the laboratory of Michael Smith as incremental changes to the technique of marker rescue. In the marker rescue procedure, a mutation in a bacteriophage genomic DNA is corrected after the mutant DNA is annealed to a fragment of the complementary wild-type DNA. Subsequently, it was demonstrated that a chemically synthesized oligonucleotide annealed to bacteriophage genomic DNA could produce a specific mutation. Unfortunately, these and other early procedures for oligonucleotide-directed mutagenesis required specialized skills and initially could be performed in only a few research laboratories. However, the procedure using bacteriophage M13 described by Zola and Smith made it a relatively straightforward matter for thousands of laboratories throughout the world to specifically and rapidly alter the DNA sequence of any cloned gene. The key to the success of the protocol developed by Zola and Smith lay in the use of E. coli bacteriophage M13, which was possible to clone foreign DNA into the double-stranded form of the virus, add an oligonucleotide with a specific change to the single-stranded form to produce a mutated DNA copy, and then recover the mutated double-stranded form in a relatively high yield. Since it was originally described, this procedure has been enhanced, simplified, and optimized, and has been used by a large number of researchers to specifically modify thousands of different genes. Random insertion-deletion mutagenesis. The technique of error-prone PCR, which is quite commonly used to introduce random changes into a target gene, is somewhat limited in the types of changes that can be introduced. The distance errors are typically introduced into DNA at no more than 1 or 2 per thousand nucleotides. Only single nucleotides are replaced within a triplet codon, yielding only a limited number of amino acid changes. With this approach, it is possible to delete a small number of nucleotides at random positions along the gene and at the same time insert either specific or random sequences into that position. This method entails the following steps. 1. An isolated gene fragment with different restriction endonuclease sites at each end is ligated at one end to a short non-phosphorylated linker that leaves a small gap in the DNA. The gap is a consequence of the fact that the 5' nucleotide from the linker is not phosphorylated and therefore cannot be ligated to an adjacent 3' OH group. After restriction enzyme digestions, disintegrates comparable sticky ends, the gene fragment is cyclized with the T4 DNA ligase to create a circular double-stranded gene fragment with a nick in the antisense strand. The nicked strand is degraded by digestion with the enzyme T4 DNA polymerase, which had exonuclease activity. The single-stranded DNA is randomly cleaved at single positions by treating it with a cerium 4, that is a ethylene diamine tetraacetic acid EDTA complex. The linear single-stranded DNAs are ligated to a linker containing several additional nucleotides, selected for insertion at one end, and the anti-mutagenesis library is amplified. Figure 8.8 is a schematic representation of random insertion by deletion protocol to introduce random mutations into a gene of interest. The inserted DNA is shown in yellow and the linker is in green. Six, the linkers are removed by restriction enzyme digestion. 7, the constructs are made blunt-ended by filling in the single-stranded overhangs using the clino fragment of E. coli, DNA polymerase 1, and then cyclized again by T4 DNA ligase. The amplified products are digested with appropriate restriction enzymes, cloned into a plasmid vector, and then tested for activity. With this approach, it is possible to insert any small DNA fragment carried on a linker into the randomly cleaved single-stranded DNA, with the result that a much greater number of modified genes may be generated than by error-prone PCR. The mutations that are developed by this procedure may be used to select protein variants with a wide range of activities. DNA shuffling. Some biologically important proteins such as alpha interferon IF and alpha, see chapter 10, and encoded by a family of several related genes, with each protein having slightly different biological activity. If all at least several protein have slightly different, if all at least several of the genes or cDNAs for a particular protein have been isolated, it is possible to recombine portions of these genes or cDNAs to produce hybrid or chimeric forms. This DNA shuffling is done in the hope that some of the hybrid proteins will have unique properties, or activities that are not coded in any of the original sequences. Also, some of the hybrid proteins may combine important attributes of two or more of the original proteins, example high activity and thermostability. The simplest way to shuffle portions of similar genes is through the use of common restriction enzyme sites, digestion of the two or more of the DNAs that encode the native forms of similar proteins with one or more restriction enzymes that cut the DNAs in the same place, followed by ligation of the mixture of DNA fragments, can potentially generate a large number of hybrids. For example, two DNAs, each of which has 3 unique restriction enzyme sites, can be recombined, shuffled to produce 14 different hybrids in addition to the original DNA. Another way to shuffle DNA involves combining several members of a gene family, fragmenting the mixed DNA with deoxyribose 1 fragments. Number 1 DNA is 1, selecting smaller DNA fragments and PCR amplifying these fragments. During PCR, gene fragments from different members of a gene family cross-prime each other after DNA fragments bind to one another in regions of high homology or complementarity. The final full-length products are produced by including terminal primers in the PCR after 20 to 30 PCR cycles. A panel of hybrid full-length DNAs will be established, as seen in Fig 811. The hybrid DNAs are then used to create a library that can be screened for the desired activity, a task that may be the most difficult and labor-intensive part of the entire process. Although DNA shuffling works well with gene families that are sometimes called molecular breeding, or with genes from different families that nevertheless have a high degree of homology, the technique is not especially useful. Proteins have little or no homology. Thus, the DNAs must be very similar to one another, or the PCR will not proceed. To remedy this situation and combine the genes of dissimilar proteins, several variations of the DNA shuffling protocol have been described. One procedure that was developed to combine the genes of dissimilar proteins and that does not rely on PCR amplification of DNA fragments is called non-homologous random recombination. In this procedure of DNAs from different sources, either defined or random DNA sequences or a mixture of both, are combined and then partially digested with DNase 1. These DNA fragments, which include a wide variety of sizes, are made blunt-ended by digestion with the enzyme T4 DNA polymerase. This enzyme both fills in 5' overhanging nucleosides and degrades 3' overhanging nucleotides. The DNA fragments are then mixed with the synthetic DNA fragment that forms a hairpin loop and contains the specific restriction enzyme sites before the entire mixture is ligated by the addition of the enzyme T4 DNA ligase to form extended mosaic DNA hairpin molecules of variable lengths. The average length of these hairpin structures is dictated by the ratio between blunt-ended DNA and the added DNA hairpins, which prevent further concatemerization once they are ligated to the ends. Finally, restriction enzyme regestion removes the hairpin loop so that the resulting sticky-ended DNA fragments can be inserted into plasma vectors and tested, and sticky-ended DNA fragments can be inserted into plasma vectors and tested for various activities. Because this process randomly recombines DNA fragments, only a very small fraction of the recombined DNAs are likely to encode the desired activity. Now, the next topic will be mutant proteins with unusual amino acids. Essentially, any protein can be altered by substituting the amino acid or another using directed mutagenesis. However, this approach is limited to the 20 amino acids that are normally used in protein synthesis. One way to increase the diversity of the proteins formed after mutagenesis is to introduce synthetic amino acids with unique side chains at specific sites. To do this, E. coli was engineered to produce both a novel transfer RNA, tRNA, that is recognized by any of the existing E. coli, amino-acyl tRNA synthetases, but nevertheless functions in translation, and a new amino-acyl tRNA synthetase that aminoacylates only that novel tRNA. A novel tRNA and unique amino-acyl tRNA synthetase pair from the Archaebacterium Methanococcus janusaki was used as a starting point for the system. The tyrosine tRNA synthetase from M. janusai, I think that's how it's pronounced, G-A-N-N-A-S-C-H-Y, is janusaki, or maybe, can add an amino acid to an AMBER suppressor tRNA, that is a mutant of its tyrosine tRNA. An AMBER suppressor tRNA is a modified tRNA that can insert an amino acid into a protein in places where the mRNA contains an AMBER codon. Excuse me. UAG, which normally codes for a stop, that is the cessation of protein synthesis, to prevent the translation fusion of proteins whose mRNAs normally code for a stop with a UAG with downstream proteins. Suppression is always less than 100% and is often dependent upon the nucleotide surrounding the stop codon. The amino acid specificity of the tyrosine tRNA synthetase from janusai is altered by random mutagenesis of its gene so that instead of tyrosine it places O-methyl L-tyrosine onto the tRNA, specifically a cloned version of tyrosine. So a cloned version of the target gene is modified by oligonucleotide-directed mutagenesis so that it contains 5' tag, 3' in the position in that portion of the DNA that encodes the amino acids that is targeted to change to O-methyl L-tyrosine once the modified DNA has been selected. It is used to transform an E. coli strain that was previously engineered to produce O-methyl L-tyrosine tRNA. The engineered E. coli strain inserts O-methyl L-tyrosine into proteins that contain a UAG stop codon, resulting in a full-length target protein containing the modified amino acid. Had the mutant gene been expressed in a wild-type E. coli, a truncated version of the protein would have been produced. This symmetry can be manipulated to insert a variety of different amino acid analogs into specified sites within proteins in the hope of producing functional proteins with altered activities. Compared with the native form, in a similar approach to this problem, researchers modified a portion of the valine tRNA synthesis gene so that the altered enzyme was able to insert the non-standard amino acid aminobutyrate into proteins. While the full potential of these approaches has yet to obturate into proteins. While the full potential of these approaches has yet to be realized, it is nevertheless clear that it is now possible to produce proteins containing unusual chemical structures and possibly having unique properties. This lecture ends here. We'll be continuing in the next lecture. Thank you.